welcome to Talks at Google. My name is Suzette, and I am beyond excited to welcome the world-renowned cardiac scientist, Sian Harding, who is here today to discuss her incredible new book, The Exquisite Machine, The New Science of the Heart. In this wonderfully written book, Sian illuminates how new scientific developments are opening up the mysteries of the heart. She discusses how the explosion of new science, including ultrafast imaging, gene editing, stem cells, AI, to name a few, has critical real-world consequences for health and well-being. Sian Harding is Emeritus Professor at Imperial College London, where she has been the head of the cardiovascular division, as well as director of the British Heart Foundation Cardiovascular Regenerative Medicine Center. Her work has focused on the myocardium in heart failure, especially beta-adrenergic mechanisms. And just to name a few of her many accomplishments, Sian was scientific principal investigator on the first UK gene therapy trial in LVAD patients, former president of the European section of the International Society for Heart Research, and board member of the British Society of Gene and Cell Therapy. She was also special advisor to the House of Commons Science and Technology Committee on Regenerative Medicine. Sian has been given a Lifetime Achievement Award from the European Society of Cardiology Heart Failure Association, and she will receive the Imperial College Medal this year. So for the audience, if you're joining us here in the live stream, we will have time for Q&A towards the end of the talk, so please go ahead and put your questions for Sian in the live stream chat at any time throughout the discussion. So without further ado, I am honored to welcome the amazing Sian Harding.
Thank you very much, Suzette. What a wonderful introduction. That's very nice. And thank you very much for having me. I'm just going to share my screen now. I'll put the slides up, I hope. And I thought I'd just start with the cover of the book, because I had a feeling that this audience might appreciate this. I gave the brief to the artist that I wanted to start off with the mechanical idea of the heart, the steampunk version of the heart, then go on to the microelectronic side, and then finally end up with the neural networks. And I think they did really quite a good job there in realizing what I wanted to do. Anyway, one of the reasons I wrote the book is because I've been working for 40 years or so on this. And of course, the reason we work on it is because we're working on heart disease and we want to understand and help heart disease. But I'm just so impressed always by, and more, even every month, right up to now, there are new things coming out about the heart, just how great the heart actually is and how many of us actually last to the end of our life, 80, 90, 100 years with this fantastic organ here. And so if you think about that, our heart beats 100,000 beats a day, so that's 3 billion beats in a lifetime. And sort of the kind of thing that would happen with your washing machine, if you're thinking about the spin cycle, you'd have to have 10 washes a day for 1,000 years to match that kind of workload, approximately. So you can see that one of the problems we have had with the heart is in the science and trying to improve a failing heart is that the heart is so very well made. It's really very difficult to beat that kind of engineering. So one of the questions that we started with was how much repair is there in the heart? How much does the heart turn over? I mean, your hair grows all the time, your liver regenerates. What about the heart? And so I started off by looking at these individual cardiac muscle cells that come from the heart. So if you think of this as being your heart, and this is a small part of your heart just cut out there, you can see that there's a sort of network, a jigsaw of these cells. And we managed to tease them apart using enzymatic digestion. Generally, when they're in the heart, they connect with each other electrically. Every cell is connected to every other cell on multiple points. And so the heart really acts as one giant cell. And when you take these out, you can put them in a bath, a Petri dish, and you can see them contract away. If they're pacemaker cells, they'll contract by themselves. If they're cells from the ventricle, you need to stimulate them, but they will contract away in the dish for hours, days, just like little tiny hearts. So the distance from here to there is it's only 0.1 of a millimeter, width of a human hair, really. So this is the normal one beating away, and this is one that's dying. And you can see, as I was saying, that it's fibrillating, and it's now hyper contracting. So this is what we call the popcorn phase. All the muscle fibers, all these little stripy things, have now contracted and buckled over themselves, and the cell has sort of burst and released its contents. And we've got about 5 billion cells in the heart, and now if you have a heart attack, about 2 billion of those in your left ventricle, the one that pushes the blood around the body, will have died. So the question was, can we make new cardiomyocytes? And it was very difficult to tell. There was a lot of argument about this, and the way we've done this is the only way that we could do is to carbon date the heart. Now, most of the time, you think about that for fossils. So we're thinking about 14 carbon, the radioactive carbon, which is taken up and during your lifetime and then released as when an organism, a tree or a dinosaur, whatever, dies. Usually the turnover of this is very slow in the atmosphere, very low levels in the atmosphere. But in the 1950s, the bomb tests were above ground for some years, and then there was this big spike of radioactive carbon. Then suddenly somebody realized this probably wasn't a good idea, and so they became underground. But if you've been born along this curve, this time, then you can examine tissues from the heart to see if your birthday and your myocyte's birthday are basically the same. And in fact, they could tell that there was just this little tiny bit of turnover, which was about 1% a year when you're younger, and it drops to some extent, but it doesn't go away when you're older. And so basically 50% of the cells you have, these cardiomyocytes, these little muscle cells, will be with you from the moment you're born until you die. So that's pretty good going, 100 years or so for that one tiny cell to carry on beating. And so I'm just going to say this quite quickly, because I'm sure you probably know this, and you can ask me in the questions afterwards if you want to know more. What things are bad for your heart? And you probably know about, well, aging is the worst thing. All the heart disease is very much a disease of age. But the usual things, smoking, being overweight, high blood pressure, cholesterol, lack of exercise, diabetes is a big one that's increasing over time in all the populations of the world. Alcohol, unfortunately, yes, there's still some evidence a little bit of alcohol could be beneficial, but basically that sort of amount is getting less and less as we find out more and more. I'm sorry to tell you that bad news. There are things that are bad for your heart you might not know about. So infection. Now, you probably have realized this because of the COVID epidemic, that people with heart disease or underlying heart conditions were doing worse and surviving less than healthy people. But that's been always true for other kinds of infection too. And so flu and things like that. So it was particularly sort of evident for COVID, and there was damage to the blood vessel lining, but it's been known about for a while. Chemotherapy. Now that's an interesting one, because it's kind of being a victim of our success. So now people who are being treated for cancer are living longer because the cancer drugs are successful. Breast cancer, for example, perhaps caught early, 99% of women will survive. But the drugs that are used to kill cancer are used to kill fast dividing cells. That's why your hair falls out, for example. But the heart has got so little regenerative power that just even knocking off a little bit of this with the chemotherapy, it'll damage it. And we knew that the early heart failure, the early cancer drugs were really poisons, and so they really do damage all the organs, honestly. When we got more specific things like monoclonal antibodies for the cancer, breast cancer, for example, thought that maybe that was going to solve the problem, but unfortunately not. In fact, and especially if you combine them with the older drugs, as in one trial, perhaps about a quarter of the people who had these new cancer drugs actually had heart failure. So there's a great deal of investigation now for new drugs that are not cardiotoxic. Obviously, the cancer killing thing is the main thing. You can't really stint on that. You've got to get the cancer first. But so some new drugs with new mechanisms of action, but also clinics in the hospital, the cancer hospitals, where cardiologists and oncologists come together to assess risks for people and treat people as they're going along, and just keep monitoring them. Pollution. I have a really interesting study in the book where, if anybody knows London, the people groups of people were asked to go for a walk, either in Oxford Street, which is a very busy shopping street with diesel. There used to be diesel anyway, buses and taxis. And at the end of Oxford Street, there's a very lovely park called Hyde Park. So it's beautiful, it's a large park, but it's still in the centre of London. So it's still not, in the middle of the countryside. And the groups of people either went for a walk in Oxford Street or later went for a walk in Hyde Park. And they're a mixture of people who are apparently healthy and people with heart or lung disease. And if they went for a walk in Hyde Park, then they even a couple of hours walk, just at their own pace, produced a measurable effect on heart health, improved heart health, blood vessel particularly, and that lasted for a couple of days. That's nice to know. Down Oxford Street, it was exactly the opposite, that you could see definitely something bad happening to the heart, even within that couple of hour window. And those two places only really had a small difference in pollution. The Hyde Park was just below the sort of regulated limit for ppms, and the other one was Oxford Street was just about twice as much. So that's a low socioeconomic status is a risk, even without any of the other things that go with poverty. And people often say that people in the low socioeconomic groups don't eat well, don't exercise enough. But even if you take away all those things, just being low in the hierarchy has a measurable effect on your heart. That was shown with civil servants in the Whitehall study. They have a very sort of ordinary job, not too stressful anyway. But those in the higher echelons had a longer lifespan than those in the lower. And even if you take a cage of mice in the lab, they've got quite nice conditions. They've got things to play with. They've got wheels to run in. They've got lovely food. But when there's a cage of male mice, the hierarchy from the alpha male down to the lowest, the one at the lower end of the hierarchy can develop atherosclerosis and a heart condition just from being in that hierarchy. Then hidden genetic conditions. So it's become very clear as we've done the sequencing how common it is to have a heart mutation. The hypertrophic cardiomyopathy mutation, 1 in 500 births. There's another one called titin, and that's one of the proteins in the sort of stripy bit, the muscle fiber, that allows the cell to relax in between beats. And it's one of the longest proteins in the body, so it's very prone to having these mutations. So here it was found when they looked at people with a genetically mediated cardiomyopathy, heart failure, about 25% of them had some kind of mutation or variant in titin. But then when they looked at, that seemed to be, that was obviously a very clear cause of the myopathy. When they looked at healthy volunteers, they found that actually about 1% of healthy volunteers had them. And so you can see this is the protein here, and these are all the mutations. And you can see the overlap between healthy volunteers, the population, just a general population, and people with dilated cardiomyopathy. You can see there's really quite a lot of overlap between those. This is just one mutation, and there are many mutations. So there are lots of us walking around with unknown genetic mutations. And what it seems to be is that you can be well for all your life with these, except if you have some other insult to the heart. So if you have chemotherapy, for example, or pregnancy can do this, or high alcohol consumption, then your heart will be very much more sensitive, and you can develop this cardiomyopathy. And then another thing that I'm often asked about is this sudden emotional shock thing. And yes, stress is bad for your heart, and particularly this sudden stress. And there's a disease, in fact, two diseases called broken heart disease. I often call this my sex, drugs, and rock and roll slide, because so many things can cause this. So there's bereavement is a really big one, and this hence the name of broken heart syndrome. So when Debbie Reynolds died just after Carrie Fisher, her daughter died. But other things, dancing, beverages, a fair, even things like a surprise birthday party can do it. And one of the diseases, one of the two of these, is called Takatsubu Syndrome, which is particularly seen in postmenopausal women. So you have many different threats to the heart, many different things that can damage the heart. But what can also damage the heart is the body's response to this. And so your body kind of magnifies this. So we're getting quite good at treating heart attacks, but the incidence of a disease called heart failure is increasing. So heart attack is something that obviously damages the heart. It kills some of these cells. But blood pressure, diabetes, etc., all of these will cause some damage that mean that your heart is not working as well as it should do. And your body senses this, and it tries to improve the activity of your heart by various compensatory mechanisms. The walls can thicken, or it can dilate, get big. You can retain water to load the heart, or adrenaline. Adrenaline will come and stimulate the heart to beat harder and beat faster. And while these things are effective in the short term, they're really emergency responses, and they will damage the heart further, and they will drive it into decompensation and this disease called heart failure, which will now is about as virulent as most of the different cancers in terms of mortality. And so just to be clear, this is the symptoms of a heart attack, and you should all know these. You should know, go straight to the emergency room, get the ambulance with the defibrillator in it. Don't sit around the GP or and wait. So you pain in the chest, pain in the left arm, jaw, or back, you're grey, sweaty, very tired, faint, nauseous. So that's the heart attack symptom. Heart failure is quite different. It's said to feel like drowning. You have breathlessness from water retention. So the water around your lungs makes you breathless. You get swollen ankles for the same reason, water retention. You feel extremely tired and faint, and particularly tired on exercise. But then even when you're at rest, eventually you feel this extreme tiredness. And this is very dangerous signs. And the reason why the body does this is because in our evolutionary past, we didn't really have heart attacks. We didn't live long enough to have heart attacks. And we also had very healthy paleo diets, of course. But the main threat was injury from something like a predator or falling over, etc. And so this is a man being gored by a bison here in the cave paintings. And so what the body is responding to is, in fact, hemorrhage. So it's thinking you're losing water from injury, from a hemorrhage, or you're wanting to run away from the predator. You're trying to the fight and flight response. The adrenaline is now activated. And so those are, as I say, great when you need to get away. But when these kind of responses are prolonged for days or weeks or years, then they damage your heart and they push it further. And so that what we do for the heart now, the things that we mainly do are first to stop these things, these damages happening more or again. So statins, blood pressure control, keep taking the tablets, aspirin to prevent clotting for myocardial infarction. So you have all these things like ACE inhibitors, beta blockers, diuretics, anti-diabetic medications, which are actually quite good for heart failure, even if you don't have diabetes, interestingly. And so we have those. And then we have drugs that will prevent the progression of disease by blocking this body response to the initial damage. And actually, they overlap quite a lot with some of the preventative drugs and the heart failure drugs. And so these will kind of turn off the body's response to that. So those are the mainstay now for what we do for heart disease and heart failure. And so there's a lot we still need to do. And so I'm just going to now think a little bit about what we're trying to do in the future, and trying to do now and for the future. And I talk in the book a lot about the tools we're using, like imaging, which I don't have too much time to talk about, but thought you might be interested in a few of the slides about the use of big data to understand more, and some of the effect of the use of AI to help with the diagnosis and prognosis of heart disease. So I'm just showing you now one of the studies that's been done using very large data sets, and there's lots and lots of studies coming out from places like UK Biobank or from very large cohorts, or from the original Framingham study that show have these big cohorts and are able to show very small effects because they have these very large numbers. Now, the UK electronic health records are just getting themselves together, really, and because we have the whole population because of the National Health Service. So this should represent a very rich data set. So this is a set of so 250,000 patients, and they've got this very common test. They've had this very common test for heart disease, which is troponin. Now, troponin is again another one of these proteins which are in the sarcomeric structure, the muscle fiber structure here. And when your cell dies and it releases its contents, then it will those things will spill out into the blood. And so when you find troponin rising in the blood, that means some of your heart muscle has died. Now, these tests are great, but they've become more and more sensitive over the years. And the ones we have now are so sensitive that they will give you a positive test, even if 40 milligrams, we did this with our cardiomyocytes, even if 40 milligrams of your heart has died. And that's a grain of rice, basically. And that's first, you'd never measure it on any imaging. And also, it's very unlikely to actually have had a significant effect. In terms of it's very hard to imagine that it could have this kind of significant effect. So there's a lot of people coming in having these positive tests, and it's quite difficult to know when they have these low levels what to do about it. So they just took patients who'd come into the emergency room and had a troponin measurement positive, and some of them were admitted to hospital. and some of them seemed to be fine and went home again. Some of them turned out to have heart disease, but a lot of them didn't have heart disease. And and that's quite common when people come in with chest pain. And some of them, then they went on to have various different kinds of management. And so the findings were really very interesting. That so the output was just death, and they didn't really divide it into death by cardiac causes or non-cardiac causes. Just one input, troponin, and one output, death. And you can see the risk ratio here. So this is, the red line is 1, and if you're above the red line, you've got an increased risk. So people who had a positive troponin test in the 18 to 29 age group were very likely to have a higher mortality if they had this troponin test. They were from 16-fold something likely to die in the later period. And even up to 90, although it's a very small change, because it's a very small change, there was a significant change. So you can see this here. If you look now, these are grouped. This is the 18 to 39, 40 to 79, and over 80 group. And so in the younger group, death rates are not high, but there's a big effect on when you test positive for troponin. In the higher age group, obviously your risk of death over 36 months when you're 80 or 90 is going to be higher. But even here, there's a strong effect of having a troponin positive test. And so this was really very surprising all around, that even these very small changes were showing this big effect on mortality. Also, that it happened quite quickly. And so the effect, the lines diverge very early, and then they stay, they stay more or less the same, or diverge a little. So when people had these tests, they would say, oh, well, let's see what happens. But clearly, this is showing you must do something very quickly, because the risk is all in the first couple of weeks. So this is big news, and has, just this one thing, one input, one output. Don't really know anything about the patients apart from that. Has given some very clear indications about what you should be doing. So here is one with another one that's been done very, very recently. And so moving on from big data to actual using deep learning. This was a prediction from a test called positron emission tomography, where you could see various things. You could see, at the same time, you can see the heart shape, you can see its function, and you can see how well it's being perfused with blood. So you can see all these at the same time. And so there's a lot of the data there, and it's been very difficult to put that all together and get some kind of sort of output as to what you should think about in terms of patient survival. So this was a deep learning thing. And here you can see that so these are the patients without very much ischemia, the blue ones. So they didn't have very much damage to their heart. These are the ones that had quite a substantial ischemia. And here you can see the scores, the deep learning scores now have really distinguished between patients. So you can see those who have a particular set of characteristics picked out by the deep learning algorithm have much greater risk of death in a 2-year period. And this was called explainable deep learning, i.e. what it did was it gives a kind of map of all these different pieces of information and targets for each individual patient what aspect of this, all the different data that was put in, was most important for coming to this conclusion. And so it's very useful for the clinician, both for predicting which patients they should be concentrating on, and which features of the patient, is it perfusion or the function of the heart, that it should be focusing on. So this is showing, and there's quite a number of other studies which have combined imaging with deep learning that have shown that some very interesting things for the cardiologist. So what do we lack now for treatment of heart disease? So I've told you we do have the drugs. We have, as you know, the pacemakers and implantable cardiodefibrillators, so that will shock your heart back into some kind of rhythm. There's been big advances in those. You can have now one that paces your heart, shocks it if you need to, synchronizes both sides of your heart, which helps a lot with heart failure, and sends all the information back to the pacing clinic every night via a box on your bedside table, which has revolutionized the treatment for patients with arrhythmic heart disease. Transplantation has improved a lot with systems for taking care of the heart once the donor heart, as it's traveling to the patient, but it's limited by the number of donors, which in fact is getting lower because safety advances in traffic and things like that. And so that we have, for example, 2,000 transplant operations in the UK, whereas you've got 900,000 people living with heart failure. We still, though, haven't got a full artificial heart, and this really speaks to the engineering of the heart, that we've never managed to do this. We've been trying since the moonshot in the 60s, the 1960s, and we haven't got to that stage yet. So we have the various operations like revascularization if you've got a blockage, giving you new blood vessels to the heart, coronary artery surgery, valve surgery. But what we lack is something that's safe to stimulate the heart. So people with heart failure, we don't have anything to actually improve heart function. We were using gene therapy to try and do this, and gene therapy is opening up many different doors. And then what we finally need, though, so this only works on the cells that are left. So it only works on the cells that didn't die. What we really need to do is to add new muscle. So I'm just going to tell you to finish up a little bit about this. And so we've been using stem cells, and you've probably heard a lot about stem cells. And stem cells are basically cells that haven't really decided what to do yet. They can proliferate and grow in an unlimited way, a bit like cancer cells, actually. But then when they are pushed by the body, they can go into liver cells or nerve cells, cardiac cells, and as you know, cardiac cells then stop dividing, and so we don't get too many of those after that. And so differentiation, this is called differentiation when they go into these different processes. And they were first discovered in the early embryo. So they're embryonic stem cells. They're pluripotent because they can make every cell in the body except for the placenta, in fact. And they, if you take them out and put them in a dish, you can change the conditions using things that we know from developmental biology and push them into being cardiomyocytes. So this is very interesting. People had problems with the embryos because, of course, what happens is if you're using a human embryo, you've basically destroyed the embryo at this point, even though it's only 10 days old. So that had big ethical implications. So Shinya Yamanaka won the Nobel Prize just over 10 years ago for showing a way that ordinary cells, so skin cells in this case, could be taken, but blood cells also, or cells picked up from urine or whatever. And if they could be grown in a dish, they could be reprogrammed back to their earliest state. So like reset the factory settings, so they became like stem cells. And they're induced pluripotent stem cells. And they will have all the same properties to change into all the cells of the body. So these are some of the cells that we made in the laboratory. So now the techniques mean that you can make about 99% of something, billions of cells, 99% of them being cardiomyocytes. They're young cardiomyocytes, they're like embryonic cardiomyocytes, but that means they're very vigorous and they're very resistant to hypoxia. And so they have many advantages over these, and they will beat away. And you can make them into engineered heart tissue, so you can make them into pieces of heart muscle. And you can, because they've come from a specific person, you can give them back to that person. And they theoretically, because we're only just starting to do this, but theoretically they won't be rejected by the person, because they'll be the same genetic makeup. And just a slight detour now, before I go talk about that, they will also, if you have any kind of mutation in your body, you can, if you put them in a dish, you can see them behave as if they have this, to show that the effect of that mutation. So you can kind of look at the heart of a person without actually taking out their heart. So here, for example, this is the genetic arrhythmia. There was a patient, there's 76 patients from some Spanish families that had this particular mutation that produced myopathy. And they tended to have very arrhythmic hearts, the rhythm disturbances, and they would die from these rhythm disturbances. And so we took cells from three people: the father, who was a carrier of the mutation, one son who was a carrier of the mutation, and one son who was not a carrier. And we made them into iPS cells, and then we made those into cardiomyocytes, and we then made those into engineered heart tissue. And so this is about 1 centimeter long. This is the heart tissue beating against some posts. And then you have the normal subject, and that's beating quite regularly. If you look at the one of these, this is somebody with a mild mutation, and you can see that every now and again you get a little double beat in these, and so this is a mild kind of arrhythmia. And then if you look at this one now, you can see that this is a very severe arrhythmia. This is what you would call fibrillation. This is where you have to defibrillate somebody, because really it's not beating in any coherent way at all. And when we had these cells, we could edit out using gene editing. We took out the mutations from these two carriers, and we could see that stopped the arrhythmias. And we put, conversely, we put the mutation into some normal subjects, and we could see that now that produced the arrhythmias. So this is an incredibly powerful technique to understanding what's happening in your body with the different mutations. But going back to the patient-specific repair, now we can take this and we can make patches for the heart. We tried injecting these directly into the heart muscle, but because it disrupted the substrate of the heart, disrupted the wonderful connections between all the different heart cells of the heart itself, they started to disturb the rhythm of the heart. But so what we've done is to take them. This was in a rabbit heart now. So we've made a patch that's about the size of my thumb, and put it on the heart where the damage from the heart attack would be. And on the rabbit hearts, we were able to show that worked, that there was an improvement, and the hearts were functioning better after that. And so that's been scaled up. It's easy to scale up this technique. So here, patches have been made that are the right size for human hearts. And so here we have this is got about 500 million cells in, and it's probably about the size of your palm, 7 by 5 centimeters. And these have been used now in very early studies. So here we have the first trial actually used the embryonic progenitors, and they put it in under the sort of lining, the skin that lines the heart, the pericardium, and slipped it in under there in this patch form, and that was safe. Then in 2020, people started using the iPS-induced pluripotent stem cells. This was done by the unit where the Yamanaka worked, that first made the iPS cells. And they've just, in fact, published in August this year the first results out of these, and showing, I mean, these are very small studies, but they're showing some improvement, showing that it was safe, basically. And so we are there. This is where we are at the moment with these different cells, and there are studies going on in France and going on in Germany now to increase this. So I'm going to stop there because I know that I need to. We had a little delay, and I need to get back to questions.
Good. Thank you so much, Sean. So first off, what an incredible lecture. Thank you so much for that. I mean, engineered heart cells and where we are at now with that research is just kind of mind-blowing, really. And so just a quick reminder for the audience, if you have any questions you want to ask, please do go ahead, drop them in the chat. But I will kick off, and I just would love to jump right in and talk about your new book, The Exquisite Machine. So first off, I absolutely love the way the book is written. So speaking for those of us who are not career scientists, you really do a fantastic job at presenting extremely complex topics in a really engaging way. So first off, how did you come up with the idea to write a book like this?
Well, one of the things is, as I said, I've been continually surprised and overwhelmed by what we find out about heart. I just felt people didn't know this, and I wanted to convey it, and I wanted to try and write something that people could read without wanting to poke their eyes out, basically. So the scientific writing is so constraining. It's like writing an equation. You have to be absolutely precise and exact. Can't use an adjective. I tell my students off if they use the word vast. No, that's far too emotive. So I wanted something that I could put some adjectives in, and at least say amazing or something.
I love it. Well, you have succeeded in that, and honestly, I do wish that you authored the textbooks I had, throughout studying different aspects of science, and I wish we could have more of those types of conversational language. Let's talk a bit about your phenomenal career. So in your book, you express, once upon a time, I fell in love with a cell. Fantastic quote. Can you tell us more about that? What led you to devote your entire career to cardiac research?
Well, partly I was choosing my PhD, and I could choose between. They said you could have anybody in the department. It's a pharmacology department, so they had people doing all sorts of things. And I would have liked to have gone into the mathematical side, but it was very crude there. I mean, it was basically partial differential equations, and that was it. And it was then the brain also very interesting, but again, extremely crude, just cutting up brain areas and just putting them in a test tube. So the heart at least had some really fantastic imaging even then, and so I could see that there was something you could do with that. And the other thing was that, in fact, my father-in-law at the time had a heart failure, so I could see I was had an immediate emotional sort of connection for me to do something about the heart.
Wow. And can you describe a bit about what it's like to actually work with heart cells that you created? Like, the way that you've described it is once again very mind-blowing. So I can only imagine actually actively working in a lab with heart cells that you've created must be quite the experience.
It is, and of course, when we started with the embryonic ones, and in fact the mouse embryonic ones, and it was just that somebody who had some that had showed me. She said, "I can see a tiny beat there," and you could just see a flicker, tiny flicker. And what they were doing was they were just letting them run without pushing them one way or another, and they were making something called embryoid bodies, which was basically like all the cells mixed up. And so there was just a few heart cells in there, and I could just see. And having used these adult cells for, well, up to 25 years up to then or something like that, they're great, but they have their limitations. First, human. I wanted to work on human cells whenever I could. You have to reduce animal work sometimes, but I wanted to use. So you have to go and stand in the operating theatre, and for the night while people are doing the heart transplant, they often forget about you because clearly you're the least important person in that room. And you're trying to.
I mean, I don't know about the least important, but
seriously, the patient is the most important person. Then the surgeon.
Fair enough.
So right down the list there, it's the person waiting for a piece of tissue for research. Take it back, work on it, and I worked out the way to get these cells out, and also how to measure that contraction. So I was sort of ahead of the curve there. But they do, once they're in the dish, they will last for some hours or days. But what happens is, because they're out of that structure of the heart, they kind of die of something like this can be called anoikis, which translates as homelessness or loneliness. And because they don't have that sort of push and pull all the time, they just atrophy and fade away. And so to have something like this that you could make yourself, which was human, which could be matched to a human. Obviously, we made some from our own, cells. Why wouldn't you? And it could go on. They go on for a year or so. In fact, you just, it's how much you can afford really to keep them going. So we've had some dishes where we just had some little clusters of beating cells, kept them at the side, and a year later they're still going, and we had a little birthday party for them, as I've said.
Wow.
Etc. But it's just comparing those two things is just incredible.
That's amazing. Thank you for sharing all of that. So you cover a lot of ground in your book, and one of the topics that you mentioned in your lecture and is very close to everyone's heart at Google is the topic of big data. So you devote a whole chapter to this, and I guess just for the the kickoff here, why is it important to incorporate big data in research and understanding disease?
It's like having another kind of microscope in a way. You can see such small effects when you have a large number of patients. And I start off with the genetics, where it's very obvious that so you have some mutations in the heart, but you have another thing called the polygenic risk score. So for something like coronary artery disease, there isn't one mutation. There isn't. So it's a lot of very tiny things happening. And if you have what was happening, people would have 10,000 patients or something like that, and they'd come up with one set of genes, and then another set would do the same, and with different patients come up with another set. Clearly, there was something going wrong. It wasn't until you could got hundreds of thousands or millions of patients you actually get robust enough data to do it. And it's the same with this. I talked about the electronic health records. With the Titin mutation, they took the normal volunteers to see if they could see anything that was different about these hearts. Could see some very subtle changes when you combine the imaging and this sort of deep learning type of thing to get these really very deep data sets analyzed. Many different things. I talk about the effects of having a male cardiologist treat you if you're a woman rather than a female cardiologist. Now, there's anecdotal evidence for that, but until you've got this analysis of 1.3 million heart attacks, you don't get this hard data that a woman is twice as likely to survive if treated by a female cardiologist.
Wow.
So you can't argue with that stuff.
And I mean, talking about the scale of data that you need to kind of get those insights, how challenging was it, throughout your career? The evolution of technology, I'm sure, has helped in many ways. And you touched upon AI as, one of those areas. But how helpful has that been, and how has that changed throughout your career to be able to analyze such vast swaths of data that are critical to really understanding and identifying those patterns?
Well, of course, we sort of knew that this was happening, but so that when something changes in your heart, when it's not one thing that changes, you go and you say, I'll look at titin, I'll look at this protein and what it does. And you know, we'd always do these experiments where you'd knock down a protein or upregulate a protein to try and find out what it does. And so you're looking at one bit of the jigsaw at a time. But actually what's happening when you have a stimulus to the heart or a damage to the heart, many things change at once. You know, and so you will have to have a systems approach to understand all the things that are going up and down. And with time, it changes from the earliest time to the latest time. So if you know, all those things were there, but we just couldn't look at them.
Yeah. Wow. Before we move on to audience Q&A, because I believe we have a couple of questions for you, I would love to ask, what do you hope that people take away from your book?
Well, I hope that they just don't think of the heart as always something that's diseased. That's all what I'd like, you know, that it's, of course, as I say, it is, and that we have to think about that.
Sure.
But to understand that the fact that it has evolved so perfectly to do what it does means that most of the things we do are very difficult. So we've never managed to do this whole engineering thing. As I say, we've got some things like left ventricular assist devices that have helped. When we try, for example, you know, it's always been quite a mystery why the heart doesn't regenerate itself. Why should your hair grow? All this hair, well, you've got to keep cutting it all the time, and then your heart, which you've only got one of and you really need, doesn't do this at all. So after a lot of experimentation, worked out how you might make, not me, some other people, worked out how you might make heart cells divide more quickly. And so the first experiment to do this worked in the smaller animals, went to a pig to work out whether it was going to work before it went into human, which is essential to do. And so found that, yes, worked brilliantly. Put in a gene therapy vector to make the cells divide a little bit faster. And with a heart attack, it repaired itself. But so about 2 months later, the animals got these very bad arrhythmias and died from these arrhythmias. And what was happening was that too many cells were dividing. And so when a cell divides, it has to break its structure down, divide, and then build its structure up again. So if too many of those are happening, doing it at the same time, the heart can't stand it. It's disrupting its whole jigsaw, really. And so now we understand why the heart doesn't regenerate.
Wow. There's so much to learn. All right, let's jump to an audience question. All right, so the first, for heart repair, is a biological regrowth solution looking like the right path going forward? Or is there still space for trying to create a mechanical replacement for a failing heart?
That's a good question. It's good to have the competition between the two. It's the sheer, with the heart, the sheer consistency that's required. I mean, when you think about, you've got the right and the left side of the heart, and they have to, so the right side goes through the lungs, left side goes through the body, and they've got to be putting out exactly the same amount of blood. This is also very important, because if it's even different by 1 teaspoonful per beat, then that's 500 liters by the end of the day. So, you know, you do the math, do it again. So I hope I've done it right. But no, you have to have that so perfectly done. And so what's happened is that the partial ones have got round this by only spotting, for example, the left ventricle. And they've started off with trying to do the pumping action. But that, again, the mechanics of that's very difficult. You have to have a compressor, you have to have a line, all those kind of things. And so what they have now is an impeller system, and so you get somebody without a pulse because it's just sort of like an archimedes screw type of thing, and it takes the blood from the bottom of the heart, feeds it into the aorta, sort of partially bypassing the heart. But even that, the blood reacts differently. It breaks down with the impeller, and so you get some problems with blood products and things like that. So it's very tricky, that kind of engineering. The regrowth one, though, I've just told you the problems with stimulating the natural growth. So obviously you can't go too far there. It's still being pursued, that angle, by just pulling back, having something a little less strong, or something that's time limited, so you can turn it on and turn it off again in a controlled way. So that's still a possibility. The patches, so you can't inject it, but the patches, that's still going to be, I think, a possibility.
Great. All right, let's do another question. All right, thanks for the fantastic presentation. Do you think it's feasible for iPS cells to be 3D printed into complete transplants? It's being done with liver cells, I understand.
Yes, so that's a really interesting question. First, I think it's possible. The heart is helically wound. If you boil a heart, interestingly, you can unwind it, and it'll wind flat. So yes, don't try this at home. But so you have to get that helical thing. So it's a ringing motion that it does, rather than just a pumping motion. And so you do need to do that. But it's the blood vessels, getting the blood vessels in, that's been particularly tricky. And so you can 3D print quite large blood vessels, but the micro capillaries have been a tricky thing. The capillaries, in fact, form spontaneously. When we put our patches on the rabbit heart, we found these little capillaries running from the rabbit heart into the human patch. We could see the blood going through into that. They don't have enough width for the big blood flow. But I think what the people are trying mostly at the moment is to try and 3D print large ones and get them to join up with the spontaneous small ones.
Wow, that's really cool. Just a small project, of research. All right, we have time for one more question. All right, great to see you are using AI for mortality prediction from PET/MPI. What other areas do you think AI can help in your work and research?
And so there's a lot of things where very large amounts of data are coming out, and where we really need this kind of thing to help. So, for example, the human cell atlas, where the individual cells are being taken out of all tissues, but the heart, and the gene products in each individual cell analyzed. And so you can see it with one cell different from another. And so the sheer complexity of that, with time, with space, and the numbers of cells, to understand how one stimulus can produce one effect. We will need AI to do that. It's just too much for that.
Awesome. All right, Sian, thank you so much for writing this book, and also for sharing your vast knowledge and your perspective with us today. We're really grateful that you were able to make the time. So once again, Sian's book is called The Exquisite Machine, The New Science of the Heart. It is available now, so make sure you go out and get a copy of this phenomenally interesting and inspiring piece of work. Thank you again.
Well, thank you very much for having me again. It's been a pleasure. Thank you.
Definitely. Thank you.